Hello, welcome back. So as we continue through chapter 3, the next section in the chapter is going to be talking about extracellular structure. So we're going to start working our way kind of from the outside of the cell inward until we can take a look at some of those organelle or structures inside of a prokaryotic cell. And we are going to focus just on prokaryotes in this chapter. So you do not have to review or learn or cover any of the material on eukaryotic structures because that was all covered in the prerequisite by 211 course. You are more than welcome to review those sections, but it's not going to be any testable material or assessed material. It's all going to be focusing on these prokaryotic structures. Okay, so we're going to start off with a glycocalyx. So if we take a look at the word itself, glyco means sugar and calyx means cup. So we're really calling it a sugar cup. But it could be made up of carbohydrates, which is the sugary part, or proteins, or a combination of both. So there's a little mix and match in glycocalyces. When we take a look at the different forms a glycocalyx can take, there's two options. You can have what's called a capsule or a slime layer, which I always think is a really cool micro word. So the major difference between these, and it could be their molecular makeup as well, but structurally, a capsule is more organized and kind of clean looking, if you will. And a slime layer is more disorganized and just kind of flowy and not quite as secure, I guess, would be the comparison between a capsule and a slime layer. So here, this first picture is showing a electron micrograph. So the plasma membrane and the cell wall would be right here. That's the dark line. And then all of this stuff right here, all of that would be the glycocalyx, this stuff that's kind of sticking off the outer part of the cell wall. In staining, here is a micrograph of a capsule stain, and you'll don't think we're going to be doing capsule stain in this lab kit, but a capsule stain, basically what you'll do, or what you would do in a capsule stain, is you stain the background like a darker color, and then you stain the cells a darker color, but both of those stains don't stain the capsule. And so what you're left with is a dark background, you have a kind of whitish capsule, and then you have the stained cells. And so it allows you to see the structure of the capsule with the dark background, the dark cells, and the clear or the white appearing capsule. So it's a structural stain technique that you can do. So what is a glycocalyx good for? Well, one, it helps the cell from drying out. It's called desiccation. So it prevents desiccation so the cell doesn't lose water, which is required for all metabolic reactions. Notes here. It's in certain organisms that they have capsules, and not all bacteria have them. So some have capsules, some don't. It's just part of their characteristics for that particular strain or species. But organisms that do have a glycocalyx, especially a capsule, increases their pathogenicity, the ability to cause disease. And the reason why is because these can be sticky, and so they can help the bacteria, the pathogen, actually adhere to a surface. We brush our teeth twice a day, hopefully, because the bacteria that live in our mouth have capsules and they stick to our really smooth enamel. So a bacterium without a capsule would not likely do a good job of being a pathogen in our mouth because it can't stick to that really smooth enamel. But the bacteria that can cause cavities have capsules and they stick to that really smooth surface on our enamel, and that allows them to degrade our enamel and cause cavities, which is the disease of breaking down your teeth integrity. And then lastly, it also, if you are living in a multicellular organism that has an immune system, usually part of that immune system is a phagocytic cell, like a macrophage. Its job to protect its multicellular self is to go around and gobble up and eat up pathogens like bacteria. Well, if you're a macrophage and you're coming around trying to gobble up something that's got a capsule or slime layer, it's going to be really hard to do that. It makes it difficult to phagocytose. So glycocalyces and capsules can actually increase the pathogenicity or the ability of the bacteria to maintain itself in the face of this immune attack. So it can prevent phagocytosis. So preventing drying out, increasing pathogenicity by being sticky to surfaces, and increasing pathogenicity by resisting phagocytosis. All right. Okay, so those are glycocalyces. The next extracellular structure we're going to take a look at is flagella. So you may have You may be familiar with flagella. We have flagellated eukaryotic cells like sperm in males, or some protists that you might see in like a pond water sample or something. Bacteria flagella are structured different. They have the same function in that they function for motility, that they can help the organism move around, but their structure is different. We're not going to compare and contrast eukaryotic and prokaryotic flagella, but we do need to learn a little bit about what that structure looks like. So a flagella is made up of three major parts. We have the filament, which is just the tail. It's the long stringy piece. We have the hook, which is like a little curved piece that comes out of the cell wall that kind of gives directionality to the hook itself. And then we have the basal body, which is kind of like the anchor, helps to anchor the flagella into this plasma membrane in the cell wall. The hook kind of gives a little bit of curve to it so that when it spins, it's not just spinning on one axis. It actually has some, it can make some spirals in the environment that's trying to swim through. And there's a couple different organizations of these basal bodies depending on whether you're a gram-positive or a gram-negative cell, and we haven't differentiated between that yet because we haven't got to the cell wall. But if you'll notice in a gram-positive, right, remember Christian Graham, he had gram-positive cells colored purple. We have these rings and the rod here embedded in this thick, thick layer of a sugar called peptidoglycan. It's kind of a protein sugar mix. In gram-negative cells, remember gram-negative cells are staining pink, they have 4 rings, and the rod is kind of penetrating through the plasma membrane, a thin layer of peptidoglycan, and another outer membrane, which we'll see in more detail in the cell wall slide here in a little bit. So those are the structure of the flagella, right? The filament, the hook, and the basal body. Some slight differences between gram-positive and gram-negative organisms, but their job is to allow for motility. All right, so let's take a look at how some of the flagella can be arranged on a prokaryotic organism. So there's a couple words that go along with flagella arrangement. There's an older edition of the textbook that kind of kept these trichus words. I'm keeping them on the slide, but I think the newer edition is just using polar and paratrichus. So of the polar arrangements, polar just means you have a flagella either on one or both sides. So we have monotrichus, polar arrangement. It's just one flagella sticking off one end of a bacterium. We have polar amphitrichus. So amphitrichus is just a single flagella coming off of each end of the organism. And then we have a polar lophotrichus, which is a little tuft of flagella coming off of one end. So whether it's monotrichus, amphitrichus, or lophotrichus, all of them are considered polar because the flagella is coming off of one or both sides. Compare that to paratrichus, peri meaning around, okay, the periphery. So paratrichus are flagella that are on the prokaryotes that are coming off of all sides. It's not just limited to one or two ends of the bacteria, it's just all over the place. All right, so that's how they're arranged. There is one special category of flagellated bacterium, and these are in a group called Spirochetes. They're long corkscrew shaped bacterium. You can see this in the colored electron micrograph and the black and white one. They look like long curly noodles. And their flagella don't dangle off the end of the cell like we saw in those prior pictures. All of the flagella, either these little flagella here, they're called endoflagella, are housed within this membranous covering called an axial filament. And so it travels along the length of the curved cell. And so all those little endoflagella spin within the axial filament. And what that does is it allows the cell to kind of corkscrew through its environment like this. So it's almost like a built-in paddle along the length of the bacterium. It's not dangling off the ends like in a lophotrichus or a paratrichus, polar or non-polar. So this is a unique flagellar arrangement found just in spirochetes. Okay, so then how would a bacteria use its flagella? So it's kind of a fun little term. It's called the run and tumble toward or away from a chemical stimuli. So in this case, there's an attractant. Maybe this is yummy food over here or something. And so it's sensing its environment, right? So it responds to stimuli. It's saying, hey, there's something yummy over there. So then it kind of wiggles around, points its nose. Bacteria don't have noses, but you get the idea. And then it spins all of its flagella simultaneously in the same direction, maybe say clockwise, and it goes really fast, and then it kind of stops and sticks all of it. It just kind of reverses, and there's no spinning, and then it just kind of figures out where's that stimuli, whereas I'm going to point to its strongest concentration gradient, and then it'll do another run and a tumble, and it just kind of figures out where it's at, and it'll run, and eventually it should be making its way towards its food. Okay. All right, the last extracellular structure we're going to take a look at, or structures, I should say, are Fimbriae and Pili. So these are both shorter than flagellum. So flagella is the longest of these extracellular structures, and their job is purely for locomotion. Fimbriae are little bristle-like hairs that kind of stick off the surface, kind of stick out of the cell wall. Find my Fimbriae. And a cell might have a few dozen to hundreds of Fimbriae. So flagella are usually smaller in number, but they're a lot longer, and their goal is for locomotion. Fimbriae, hundreds, dozens to hundreds of these little Fimbriae, bristle-like things sticking out. They allow for the sticky nature. So maybe a cell doesn't have a capsule, but it has Fimbriae, which allow it to be sticky, so that can increase that pathogenicity. It also allows them to participate in what we call biofilms. We're going to be talking about biofilms a little bit in another chapter, but biofilm is like a smattering, a collection of multiple different bacteria, not always the same species, that kind of produce this protective layer and it makes it harder to remove them, either from an environmental location or from a biological location. And so Fimbriae can help play a part in that, which also increases pathogenicity if an organism is part of a biofilm. The other structure is called a pili, and so we can see it, this kind of little tube here. It is longer than the Fimbriae, shorter than a flagella, so it kind of lengthwise fits in between those two. And it usually is functioning in a process called bacterial conjugation. We'll see that in chapter 7. So bacterial conjugation is when two bacteria of the same generation, it's not a parent or an offspring, they come in close proximity to each other. One sends out this little pili, sometimes it's called a sex pilus, and when it attaches to the other neighboring bacteria, it's like a little tube, and it can send some of its own DNA into the neighbor, and it's sharing genetic information. This is called horizontal gene transfer. We're going to talk about that in chapter 7. So the conjugation pilus or sex pilus is a structure used for horizontal gene transfer. Okay. All right, so those are our extracellular structures. We're going to move on to the next topic, which will be talking about the cell wall, and we will learn what is the difference between a gram-positive and gram-negative cell. See you next time.